Good morning or afternoon to you. Thank you for joining today's webcast. I'm Jan Reichert, Executive Director of the Antibody Society. I'll be moderator, and this is the first in a series of webinars designed to inform and educate our members, as well as the broader scientific community, about topics relating to the adaptive immune receptor repertoire. The Antibody Society is very pleased to include a group that works actively in this area. They comprise the Adaptive Immune Receptor Repertoire community, also called the AIR community or AIRR-C. Our speaker is a very active and long-standing member of the AIR community. Professor Victor Greif from the University of Oslo will discuss steps in data processing and analysis of adaptive immune receptor repertoires, best practices, pitfalls, and future directions. Please note this webcast is being recorded. We'll take questions at 2 points in this 2-hour webcast, about 55 minutes into the presentation and then in the last 5 minutes. Please add any and all questions to the Q&A box in the viewer. We may not get to all of the questions, but Professor Greif has kindly offered to create a Q&A document after the webcast. A link to the document will be sent to all attendees. Without further ado, I turn the show over to Professor Greif.
Thanks so much, Jan, for the introduction. And hello, everyone all over the world. My name is Victor Greif, as was just introduced, and I'm the head of the Lab for Computational Systems Immunology at the University of Oslo. And this is today an AIR Community and TABS webinar on AIR data analysis. So a brief disclaimer is that this webinar is meant just as a brief overview of the adaptive immune receptor field, short AIR. So if you want to know more about any of those points that I mentioned, please consult the cited citations that I put on each slide. I may go quite fast because we have to go a lot through because the AIR field is quite large, but I put a lot of citations on every slide, so it should be quite easy to read further in all dimensions. And also, if you're not following me on Twitter, please do so. So I will give a brief introduction to the adaptive immune system, which is, as opposed to the innate immune system, the part of the immune system that records each immune event over our lifetime. So whenever we encounter bacteria, viruses, cancer, or any of these kind of things, immune information about these events is recorded in our immune memory, which you see on the right, and these are our B and T cells, which have B and T cell receptors, or together called adaptive immune receptors. And together they form our immune memory, kind of like a USB stick, which I'm showing there. So we can think of T cell and B cell receptors as natural diagnostics and therapeutics. So they record past and current immune status. They are very important for vaccine success. So not only antibodies, but also T cell receptors, as has been shown more and more now. And of course, from the pharma point of view, monoclonal antibodies, T cells and CAR T cells have become blockbuster drugs over the last years and decades. So we can use the natural sensing ability of T cell and B cell receptors for diagnostics and therapeutics research. And I guess for all of you who are in this field, you're either working on understanding the immune system from a diagnostic point of view or from a therapeutics point of view. One complicating factor about the adaptive immune receptor repertoire is that the diversity is huge, larger than 10 to the 13. We have recently summarized key advances and challenges of immune receptor analysis. And over the last decade, so since immune receptor research has started 10 years ago, a bit more, there has been a lot of progress on each of those major blocks, which are genomics, going to proteomics, computational immunology, biotechnology. So we can now high throughput sequence immune receptor repertoires. We can look at the serum antibody repertoire. We can, to a certain extent, predict antigen binding. And we have also established, to some extent, CRISPR-Cas immune cell editing. However, major challenges still lie ahead, such as ultra-high throughput antigen-specific sequencing from an experimental point of view or from a computational point of view, predicting antibody-antigen binding with very high accuracy. And I will touch on some of these points that I mentioned here throughout my presentation. So this is the outline of the webinar, and after the second topic, we will make a short break where we can answer questions for 5 minutes. And as was just said, if we don't get to all the questions, we will answer them in writing after the webinar. So antibody and T-cell diversity is generated via VDJ recombination. This is one of the fundamental mechanisms that one should know if one is deciding to start in this field or if one is working in this field. And what is happening here is that 1V, 1D, and 1J gene usually are recombining to form the variable domain, and this is true for both B cells and T cells. And this is one way of diversifying immune receptors or for getting this high diversity. B cells have one further dimension which is somatic hypermutation. Although there are some eLife papers, I think, which have also shown somatic hypermutation in T cells in sharks. But in humans, the hypermutation part is B cell specific. So if we zoom in further into the variable region, we have framework regions and we have complementarity determining regions. And especially the complementarity determining regions 3 or CDR3 has been shown to be most important for antigen binding. And all the CDRs are usually mutational hotspots. The framework regions are usually less mutated and more important for the structural stability of the antibody. I'm putting also 2 references there at the bottom, which have recently shown for T cells that a lot of the times for the beta chain, there seems to be not a D gene involved or D segment involved in the recombination. And also for B cells, it has been recently shown by Jana Safonova in Genome Research that there can also be VDDJ recombination. So, and so it seems like anything is possible in adaptive immunity. If we zoom in on the right side of the slide on the immune receptor repertoire, we can zoom in on the antigen-specific repertoires, and there one of the outstanding questions is what is the diversity so how many receptors we have for a given antigen or even a given epitope. I think for some antigens we might know this a tiny bit, but overall this is a very unresolved question, and we also do not know how the motifs look like that predict antigen binding. And we will touch upon all of these things again in the next slides. One thing that I also want to stress is that many people in the B cell receptor field mostly work on B cell receptor sequencing, so on the DNA or RNA level, whereas there's also the antibody side, so the proteomic side, because antibodies are proteins, right? So, and in order to get to the sequence diversity of the circulating gut or serum antibody repertoire, people have used proteomics. And this is also still an up-and-coming field, I would say. So there haven't been a lot of papers on that actually marry the genomic side and the proteomic side of the immunoglobulin repertoire analysis. So this is just a side comment. So, but we will mostly focus on immune receptor sequencing. Here you see the general workflow. You start from a model organism. You isolate your cells of interest, you perform library preparation, and then pre-processing, and then statistical analysis with regard to the research question that you have. The good thing about AIRR-C, which is also a short for adaptive immune receptor repertoire sequencing, or high throughput sequencing of adaptive immune receptor repertoires, allows us so to look at the central principles of adaptive immunity. So we can follow cells over time as they mature through B-cell ontogeny, and we can also look at on the right-hand side, clonal architecture, convergence selection, lineage dynamics, and evolution, and clonal expansion. So all of these things can be computationally looked at if we analyze when we analyze immune receptor data. So many of us might not have the means or sometimes do not want to generate their own data. So there are many, there are many public databases that have a lot of sequenced immune receptors. On the left side, you see antigen-specific databases. Some focus only on T cells, some focus only on B cells, some have epitope information, some do not. Some only have COVID information, for example. So all the citations you see on the left. On the right-hand side, you see citations for databases which have a more broad scope, which a repertoire scope where you can find billions of sequenced receptors and also entire data sets, so you can then go there, reanalyze these data sets, or take these data sets to supplement your own data sets for validation purposes, for example. So that's very nice. And here, I'm not going to go into detail, but this is from a paper from Mike Shugai, where he outlines what you can do with antigen-specific data on the T cell side here. So for example, you can look at the antigen exposure imprint, immunogenicity prediction, tumor-specific TCR prediction, and so on and so forth. And it's all very well explained in this review paper. So something that I think is also very important to know is, where do you go if you have questions? Because when you start, you probably have a lot of questions, and reading reviews is sometimes not enough. So we recently opened a Slack channel. There's also the BTCR forum, which is used a lot. Also, if you want to post positions, for example. And then we have also many videos on our YouTube page. And we also retweet a lot of the AIR papers coming out from our Twitter profile. Also, please follow the @AIR community profile if you are not doing that already. And please also subscribe to our YouTube channel. And please also subscribe to our Slack. Yes, so this was just an introduction that saying that we can now investigate adaptive immune receptor repertoires at high throughput. It can be performed at the genomic or proteomic level for B cells, and we can resolve the central principles of adaptive immunity. And there is already a lot of public data out there for starting your own analysis journey. So now we come to the second topic, and after the second topic, as I said, there will be a 5-minute or so Q&A. So we will talk about AIR correction and standardization of AIR-C data. And if you focus on the left side of the slide, adaptive immune receptor data, there are many pitfalls and many directions you can go. So it goes from deciding on what cells you want to isolate, if you want to focus on DNA or RNA, how you want to perform the library preparation, how to perform data preprocessing, and how to perform data analysis. And each of those things can add variability to your analysis. That's why it's good to always try to be reproducible to the extent possible. So the AIR community has been working a lot on standardization of immune receptor data generation, and also analysis, and also deposition of data. So here are some papers. So, and this is mainly work done by the working groups, which either focus on, depending on their purpose, on standardizing data formats, software, or even experimental standards, such as Bcrs standards, for example, which is still in the works. So if we look at, so if we start our journey now, so from the cell point of view, so from the cell isolation, this is just to show you that the adaptive immune cell diversity can be subdivided into many different ontogenetic stages, both on the T cell side and the B cell side, and your data will depend on what, or your conclusions will depend on what cells you take out and what cells you try to look at. Most of the studies only look at PBMC, so a mixture of many of these cell types, and sometimes then it's very difficult to then pinpoint to one specific cell type then for a specific phenomenon. So it's mostly a good idea to focus on specific cell types, and but that of course depends on the research question. Of course, focusing on specific cell types also makes your analysis much more expensive and much more high dimensional. Another, if we focus on the right side of the slide, another way to look at the data is either from the DNA or RNA side. One thing, for example, to consider for if you work on the DNA for B cells, it is then not possible to look at isotype from the sequencing point of view. Of course, you could have sorted before based on isotype, but you cannot resolve the isotype via sequencing because of the splicing from going from DNA to RNA. So all of these small things are important to take into account. Also, for example, plasma cells have more RNA than naive B cells. So if you mix them together, the plasma cells might be overrepresented in your data set. So all of these small things one should think about. A very contentious topic in the AIR community is how to define clones and what is a clone. So I will not add to the controversy here. I will just tell you what people have been working on. And I think an excellent review on this topic has been written by Uri Hirschberg, which the reference is on the left. So if you want to read more, have a look at that. But basically how people do, so this is mostly done for B cells because they hypermutate, and you want to group those sequences together that come from the same naive B cell. And all of the subsequent cells that come from this naive B cell, we call them clonal lineage. You can group those sequences together, for example, by using sequence distance cutoffs, which you see on the upper right, based on some Hamming distance. Before, it's usual to group your sequences by same V and J, and sometimes also same CDR3 length. Recently, there have been published more sophisticated approaches, mainly by the Kleinstein lab, where they use some kind of clustering to find a good cutoff for deciding which sequences group together in a clonotype and not. And also they have been also K-mer-based approaches also by the Kleinstein lab for building clonal families. So all of these things should be taken into account as well. And there is no right or wrong answer. But if you want to make big claims on somatic hypermutation or clonal expansion for B cells, it might be a good idea to look into these, how to assign clonotypes. And this, I think, is a good way to start by looking at this slide and what people have published. Another very important thing that is also a very important topic for the AIR field is sampling. As you know, the diversity of the immunoreceptor repertoire, even within one person, is so huge that it's almost impossible to sample completely in your data analysis. And you can see that by, for example, biological sampling, where you just, for example, take 2 different blood draws, and on the upper right you see the clonal overlap of 2 different blood draws based on TCR sequencing, and you see that it's not a lot. And if you were to look at biological replicates also nowadays, even with higher sequencing depth, you would be surprised at how low the clonal overlap could be. So this is something to take into account. And I think it's a bit unclear how to resolve this variability in our data, because of course sequencing takes a lot of money. So making all of these replicates without a clear way of how to use them in your data analysis is a bit tricky. And there is no real approach for that yet, in my opinion. Then there's also technical replicates, which look at which technically is a resequencing of a library, or also splitting at the PCR step and then doing things separately. And even there, you would be surprised how high or low the overlap can be. So if you have never looked at this or done this, I highly encourage you to do this, just so you see how reliable or not immunoreceptor sequencing is. Yes, and you can look at the how good your sampling coverage is by using these species accumulation curves, where you look at the percentage of reads and then how many clonotypes you have covered by sampling those reads. And most of the time, your curve will not look like this, but mostly probably, you can see my cursor, so it will not level off. It will be probably in the middle of the curve. And this means that you have undersampled your data. But probably close to 99% of all studies have undersampled their data, so you would be just +1 in the club. And you see even from this Nature paper from 2019, where they really over-sequence a lot, you see that there is not really a leveling off of those accumulation curves. What is very easy to get is what you see on the right, is a high correlation between the replicates of the V gene frequency. So that's very easy to get. What is very hard to get is high clonal overlap between the replicates. So that would look probably much worse if one would look at these data, which is not a reproach for this data, it's just how sequencing works. And this has been also shown very early on by showing that you can the deeper you sequence, the more public clones, the more shared clones you find. So saying again also, more sequences is always better. So because I asked a lot, how much do we have to sequence? The more, the better. And it's always good to know the number of cells you put into your reaction, and then it's a good estimate to over-sequence by 5 to 10 times of the number of cells. So if you have 1 million cells, it's good to have 10 million reads, for example. Yes. This is from a very old paper of us, where we showed how one could establish reliably detected clones. And I will not go into detail, but the more important thing is seen on the right, where we show that even if you get high clonal overlap between replicates, getting correct clonal ranking, so meaning that you want clone 1 in sample 1 being also clone 1 in sample 2 based on read frequency, requires even further sequencing reads. showing again how difficult it is to get really high reliability of your data. And this we have shown here, and this has also been shown by Rachel Bedford Rogers also in 2015, also in BMC Immunology, where she also shows that, so this plot L here, where that accurate clonal ranking really requires a high sequencing depth. And you see that the ranking gets better as she increases the number of sequencing reads, it really requires a lot of reads. Yes. This is also one of the few papers where people looked at the overlap of DNA sequencing and RNA sequencing, and here they found very high overlap or correlation, but there are very few papers that have looked into this. So, in my opinion, so this would be something I think for further studies would be very nice to look into, because it's quite an old paper, and now we have new technologies and new ways of looking at the data, new pre-processing suits also that are much more sensitive now. A very nice paper also coming again to how data can vary across different sequencing methods has been recently published in Nature Biotech in 2020. And here I think the main takeaway again was there was variability across different approaches, and the variability was then decreased or increased by using UMIs, so unique molecular identifiers or not. I will go a bit further into detail on the next slides on this. And I think a clear conclusion of what sequencing protocol to use for TCR sequencing in this case could also not been given, because it really depends on your research question. So we now come to the UMIs, which is also a very contentious topic in the AIR community. So I will also not light more fire here or put more salt into the wound. But the main idea of unique molecular identifiers is that you want to kind of get rid of sequencing and PCR errors, which can happen a lot and which can also bias your data into one direction or the other. So how this works is shown on the upper panel. Is you tag, so you put a sequence tag on the cDNA, for example, and then in the data analysis step, you look at all the sequencing tags or UMIs that tag the same cDNA, and then you merge them together, building a consensus read. It's what it's called. And of course, you can only build consensus reads if you have more than 3, for example, UMIs per transcript, which, as you can imagine, my usual sentence now will further amplify the need for even higher sequencing depth. So what this, so here, this was a paper by the Sci-Ready lab, where they looked at how data looks before UMI correction, where you see that one clone, which was a spike in, where you knew the sequence, the exact sequence, so here called Christina W, which had 24 false clones, and then was then corrected to one clone thanks to the UMI correction, to the consensus read building. We have also used UMIs in a completely different setting, not for error correction, but for checking contamination. So a lot of the repertoire field is focused on looking at public clones, so the overlap across different individuals, And sometimes it's not clear if something is contamination or not. And so people usually use for clonal overlap either either the CDR3 or the entire VDJ sequence, and the CDR3 is usually a barcode, but the UMI is also a barcode. So what we did here is we used UMIs to check for contamination. And how do we did this? So we looked at the CDR3 overlap on the bottom left across different samples, and then we checked whether any of the overlapping CDR3s also had the same UMI, which would be highly unlikely, which should not happen if you don't have contamination, and which would happen if you have contamination. So CDR3 that are overlapping and also have the same UMI are most likely contamination. And here we could exclude the contamination, for example. So I haven't seen so many papers using that approach, but I think it's a very nice way of using UMIs. Yes. The problem, so now this was now the good side of UMIs. Now we come to the bad side of UMIs. The bad side with UMIs is, of course, also that you can have different errors also in the UMIs, just as you have for the sequencing reads. So you can have sequencing error there as well. But the most important problem with undersampling and UMIs is that you have to have at least 3 UMIs per transcript to build the consensus read. And that means you have to over-sequence a lot. You see this, for example, here in this 2013 paper, where most of the reads had only 1 UMI. So I'm talking about the upper left plot now. And then so maybe 20% of the reads had were in groups that had 3 or more UMIs, which means that most of the data here could not be used for UMI correction. We have also seen this in our data, where I show here again the CDR3 overlap on the right, where the expected overlap, if we do not correct for UMI, is about 14%. And if we use UMIs, it goes to 0.23% because we have to throw away so many reads because we cannot use them for consensus read building. And the same has also been put into words in the Nature Biotech paper, which I'm quoting on the lower left, where it stated, yes, UMI-based methods can be more accurate, but they decrease the observed TCR diversity. So I think a takeaway for UMI is if you have a very low sequence diversity, then UMI is good. If you want to look at the diversity of an entire repertoire, then you would have to over-sequence so much that it's economically almost not feasible at this stage. Yes, so this is something really to keep in mind. What also is to keep in mind for any annotation, and I think this problem has not been solved so far, is that the D region annotation is quite unreliable. And that's why most of the time the D region is not even shown in any annotation plots, for example. So people mostly show only the V and J, and then you see sometimes a blank for the D. That does not mean that there was no D in the recombination event. Could have been, but the more likely thing is that it was just not found because the D just gets chopped up too much during VDJ recombination. So that's something to keep in mind. On the other hand, V and J assignment is usually higher than 99% accuracy for most of the tools. So that's not something that one should worry about. Alleles is something different, which I'm coming to now, is So germline gene alleles is something that has come up recently, and which is mostly a topic for the B cell receptor side, although I think people are also now working on this for T cell receptors. So what is happening here is that if we just go to the upper left of the slide, is that so before we had high throughput sequencing, most of the it was thought that there is one gene and maybe a few alleles. With high throughput sequencing and new technologies, we now know that we probably all differ by maybe one or two or several single nucleotide polymorphisms. Why is it important? Because if you, for example, look at somatic hypermutation, somatic hypermutation takes only the assigning of the mutation count is always with respect to a reference germline gene data set. If you don't take into account these polymorphisms, then for example, your mutation count might be off, or even your clonal family assignment might be off also. So that's something to keep in mind. What is interesting to note is that there are many initiatives now to look at polymorphisms, both from the AIR community side and also from the research side. So there are databases where you can look at the recent alleles that have been verified, so that you can use them in your assignment, in your clonal assignment, in your VDJ assignment. And there are also computational suits, so approaches that can infer alleles from high throughput sequencing data. Some of them are, for example, IgDiscover, which I'm quoting on the upper, on the lower right. So just go to these citations and you will see many computational approaches that can infer novel alleles or also haplotypes. So why is it important that we look at alleles? Because they are not only there for our amusement, they also have been implicated into disease. So, for example, alleles of IgHV1/69 have been implicated into better or worse neutralization of the virus, and also these also may vary by ethnicity, as many of the polymorphisms are. They could vary by ethnicity, but we still lack really large-scale data to pinpoint this. And they have also been attributed to epitope-specific antibody responses in autoimmunity. Alleles have also been, so I'm not showing this here, I'm just saying it, have also been implicated in the response to malaria, and also have been recently looked at for vaccine responses. So some alleles are more or less good in generating for a precursor B cell receptors for then generating broadly neutralizing antibodies. So this is just to say that if you're not really interested in somatic hypermutation and super correct clonal family assignment, polymorphisms is still a bit of a niche thing, but I think it's getting more and more interesting, and we find more and more of them, and hopefully with more large-scale datasets we can actually link them to disease and also antigen binding. And then it will be soon very important to build for each individual that you look at also its own reference data set, so that your somatic hypermutation count and your clonal family assignment is correct. As I said, for T cells, I don't think there are so many reports, if any, but I think many groups are working on this as well. So I would be surprised if we do not see a very high diversity on the TCR side as well. Yes, and I think I'm a bit faster than I should be, so we have then more time for question, I think. So just to summarize again, biologically, so this is, I think, a very important point. Biologically conclusive AIRR-seq really depends on deep coverage of immune repertoires. And I have been working in a lot of collaborations over the years where people thought they had that they had sequenced deep enough, and for example, UMI correction, and in the end, we could not use the UMIs because they did not sequence deep enough. So if you want to use UMIs, for example, you really need to over-sequence 10 times more that you think you have already, at least. And coverage might be assessed via replicates or, for example, species accumulation curves. I don't see so many people doing replicates, but even if you don't need them for your analysis, I would encourage you to do at least once, do them and see how bad things can really look. Then it's very important to keep in mind that AIRR-seq library preparation can induce numerous errors, such as, for example, primer bias, PCR bias. Primer bias could be, for example, excluded if you use primer-free approaches, such as race. But race might have other biases, so there's always a pro and con for everything, unfortunately. I would also like to say that computational error correction is possible using the mixer annotation suite, for example. It would be very interesting. I think I haven't seen a reported where they looked at mixer error correction and UMI error correction. I would like to see that, how good they are in conjunction or separately. Yes, so this is, and then exactly, so UMI correction seems like a good idea on paper, but might not be applicable for highly diverse samples, where you just would have to oversequence so much that you would not be able to afford it. So this is something really to keep in mind. So because some groups might just use UMIs because they think it's the right approach, and then they trim down that dataset so much that they can almost not make a biological conclusion anymore because they have lost all the diversity of the data. There exist numerous annotation tools, but care should be taken when choosing the reference genome, especially when you look at somatic hypermutations with regard to the polymorphisms. And as I said, I think a very hot topic in the future will be finding more about the functional significance, the diversity of polymorphisms and alleles in adaptive immunity, and how they vary across ethnicities, and how they're implicated in disease, and how we can use them also to make better vaccines, for example. Yes, and I think with that, I'm at the junction where we can take questions. So let's see if there are any questions.
Yes, we have quite a few, actually. So let's,
oh my.
let's start you off and get these questions answered. So first one, Sometimes multiple V genes are identified in B cells upon sequencing a cultured B cell line. Is that a technical mistake or is it normal?
Whew, that's a good question. So in one B cell line, there should be, I think, only one V gene, if I'm not mistaken. So that would be a technical error, I would think. Yeah.
Next question. Is the clonal overlap small also after the immune system has been challenged, such as in vaccination or chronic inflammation?
Yeah. Okay, yeah, that's a very good question. So I didn't show, so if you, for example, look at my Cell Reports paper in 2017, there you see that the naive B cell overlap is 14%, and depending on the antigen, it can go to 30%. But it can also just go to 14%. So it depends on the antigen. It can go up and down. We do not really understand how clonal overlap relates to antigen diversity, I would say. I think that's an open problem. But clonal overlap can usually increase if you challenge and if you look at the right subset. Yes.
Next question. Thank you for the response first. Next question. What is the best method to completely avoid next generation sequencing mistakes in the sequencing of massive repertoires? A little bit more to that question. Hold on. Strict forward reverse merging of completely overlapping reads, massive oversampling, and throwing away all those with low N.
Yeah, wow, these are very good questions. I think you can never really avoid any errors, but I think you're right, Christian, that when you so in order to minimize error over sequencing is one of the best approaches, so that you can be very liberal in throwing away the low quality reads. And what we usually also do is we throw away all the singletons just to be super strict. And this will not hurt you so much if you have over sequenced, for example. Yeah. And of course, UMIs can help if you have over sequenced a lot. Yeah. And then maybe coupling this also to computational error correction using Mixer, for example, or MyXCR. Some people pronounce it either way. Yeah.
Thanks very much. Next question. What seq depth is recommended if using UMI?
Yeah. So many difficult questions, which I try to not go into because it will result some responses from different people. But so as I said, if you know the cell number, without UMI, you should over sequence 10 times more. So then you would already have, so for 1 million cells, you should, so let's say naive B cells, you should over sequence with 10 million. So you should have 10 million reads. With UMI, I would say at least 10 times more. So you would already be for 1 million cell B cell sample be at 100 million reads. But just to be sure, one should probably go to 200 million or 300 million reads, which, as you see, becomes just impossible. But yeah, so at least 10 times more than the cell over sequencing. Yeah. So at least, so if you go from the cell, really an order like 100 order of magnitude times more reads. Yeah.
In a similar line, what are the reasons for undersampling?
Well, this is because, yeah. So this is because of the high diversity of UMIs and the high diversity of the transcripts, and you just need, and then you don't give enough sequencing reads to your samples so that you can find back all the UMIs that you have that you used for the tagging. So it's mostly people being a bit too stingy with their giving reads to one sample, and that you can talk to your sequencing facility about. You can quite finely calibrate nowadays how many sequencing reads you give to a certain sample, and that's how you can get rid of the UMI problem. Yeah.
Thank you. A lot of work has been done with custom programs because few software programs are AIR compliant. In this case, what do you recommend for code sharing and reproducibility? How much should we openly share?
How much should we openly share? I think, so in terms of software, it was the question? So
yes.
It was okay, yeah. I think it's good to be as open as possible with software, especially since it will allow other people to find errors in the code base. And because I think the adaptive immune receptors are so complex that no one software is good from the start, and many software packages that are used by the community over the past years have become better with community input, and we can only give input when something is open, right? Yeah.
Thank you. Next question. What is the importance of the sequencing length except from the sequencing depth in BCR analysis? Also, which tool would you recommend for the VDJ alignment?
Yeah. So the importance of sequencing lengths depends on how much of your Amplicon you want to cover. So, for example, for T cells, and we don't have isotypes there, and we don't have hypermutation, one can usually go quite short if your primers are, for example, in the framework 3 region, and then you just cover the CDR3 diversity. I think then you're fine, and you reasonably well can still infer V genes. Whereas, for example, for B-cell receptors, one doesn't only want to cover most of the time the whole VDJ sequence, but also the isotype. And then the 2 × 300 kit for Illumina becomes quite nice to have. Yeah, but this also depends on where your primers start in your strategy. Yeah, I think that's, and which tool you would recommend. So I think most of the tools that were suggested here in the question are for single cell sequencing. Those are very good suggestions that you give. For single or bulk sequencing, you can also use MyXCR, for example. Yeah.
Next question. If the absence of clonal overlap is a sequencing-based issue, is there a reliable machine learning model to solve this issue?
These are questions, unbelievable. Yes, no. So the answer is I don't. So I will go a bit into detail in the next part of the talk, where I talk about machine learning, and that machine learning can be on the subsequence level or on the sequence level. So to some degree, that's why people look a lot at subsequences, so k-mers, because that allows to circumvent the overlap problem. So that could be one avenue. But absence of clonal overlap, if there should be biological overlap. So if the biological system lets you expect clonal overlap, then machine learning cannot change the biology, right? But it can help to encode. It can help encode data in a different way that circumvents these things. And I will also talk about the diversity profiles that will also circumvent lack of clonal overlap. Yeah.
Thank you. Next question. What overlap percentage should we expect between mice in the case of antigen-specific B cells?
Yes. So there are not so many mouse papers, especially for B cells. There are many more human papers, and there are also many more T cell papers. So in the AIR field, people like T cells more than B cells, I think, sometimes. if you go to my paper, there is one of the figures where we show the clonal overlap between mice, and we went quite deep. So we had 1 million naive B cells, and then we saw 14% sharing between any pairwise mouse. And for the antigen-specific sharing, then we had 3 different antigens. It sometimes went up to 30%, but varied between, I don't know, 20 and 30%. So it can go quite high. Yeah.
I think we have time for a couple of more questions. Can you comment on V gene polymorphisms in mice?
Oh my God, these are amazing questions. The difficulty, so ideally, polymorphisms for one strain should not be there because, right? So that's why we use mice, because they should all be as similar as possible. That's why we use mice. However, people have shown that across strains, so for example, Bulb-C and Black 6 mice, they can use quite different V genes. And for example, the very classic immune response to NP is only seen in the Black 6 system, but not in the Bulb-C system, for example. However, if you go again to our Cell Reports 2017 paper, we looked at naive B cell CDR3 overlap between different mouse strains and found that although the V gene repertoire between these strains is different, the CDR3s were quite similar, which is surprising. But we didn't unfortunately look at different antigen-specific responses in the different strains, which would be a very nice thing to do. Somebody should do this. Yeah.
Excellent. Thanks. You're doing very well with all these tough questions. Is there a special distance matrix for Hamming distance calculation for BCRs? Because mutations that appear due to hypermutation seem to be biased.
Yes. So I'm not a super expert on on the phylogenetics and the clonotyping, so I'm not completely sure. I think people sometimes use blossom matrices to also account for some of the biological variability, but I'm not sure about special distance matrices.
Next, and maybe final question here. Do you consider clonal diversity based on VH-VL pairing or only focus on VH?
Yeah, so this of course depends on whether you use bulk sequencing or single cell sequencing. Of course, for single cell sequencing, clonal diversity should be done on the VH-VL level. I will talk more on this on the overall topic of VH-VL, but the problem I think with the AIR field in general, we all like to have single cell data and VH-VL or TCR alpha beta data. The problem with all of us is that there are very few methods that can take into account paired data for most of the computational approaches that I will present. So that's something to keep in mind.
Well, all right, we have to take one more question because she started it out saying, wonderful talk, which I totally agree. Very well done so far. How do you expect the problem of lack of clonal overlap, oops, sorry, it just moved on me, to eventually be solved in the field? Better sequencing techniques, limit sampling to diseased tissue over PBMCs or other options?
Yeah, I think, as I said, so focusing on specific cell subsets and deep sequencing is, I think, a good idea. Yeah, as a very short answer for that. Yeah.
Okay, well, to the audience, please do remember, or I'm telling you for the first time if you hopped in a little bit late, that Professor Greif has agreed to answer all the questions. So not only the ones here in person, but all of the questions here will be answered in a Q&A document that will be made available to you after the fact. Right now, we will move into the second portion of the presentation, which will also be followed by a question period. So please do keep those questions coming. Right, Victor, I hand it back to you.
Okay, so we are going to the next two topics, which is single-cell sequencing and then the computational strategies. And I will go very briefly only on the single-cell sequencing and mostly spend my time on the computational strategies. So as was already alluded to in the questions, is that a bulk sequencing has the problem that we lose the pairing of VHVL or TCR alpha and beta sequencing. So that's why single-cell sequencing as a whole is a very good idea. So single-cell sequencing preserves the pairing, so that we like. So it preserves much better the biology of adaptive immune receptors, since it also has been shown that binding can be abrogated if you combinatorially exchange the alpha or the light chain, for example. So so these chains can be important for antigen binding. Sometimes binding is preserved, sometimes binding is not preserved. It can also help us to measure transcriptome, so gene activity plus adaptive immune receptor at the same time. So to link them together, there are some papers that have done this, but I think much more research should be done in this regard. And it allows us also to be much more strict with UMI correction, since everything happens in one cell and we can be quite sure of what the result can be, although there are reports of, or people have seen with single cell data that you can have 2 VHs and 1 VL, which could be biological or not, and there it's a bit hard to correct. I'm also not an expert on this. Many people just correct these kind of data by just taking 1 VH and 1 VL, or 1 TCR alpha, 1 TCR beta, just to be completely sure that they are not looking at artificial diversity. And it could also help, of course, to obtain more correct clonal frequency ranking, theoretically, because we have paired information and we don't, so I think that's quite clear. However, what we need overall, I think, especially for single-cell sequencing, is to be able to pair it with antigen binding, even more so than the bulk sequencing. And so this is from a blog post that we wrote, but which illustrates quite clearly that we kind of need, on the experimental side and the computational side, kind of like a Rosetta Stone for immune receptor to antigen binding mapping. And there have been a few approaches, both from companies and also from academia, to kind of solving experimentally the problem, because what we think I need is much better and much more high throughput and higher quality data that maps an immune receptor to its antigen, or even better to its epitope. One can only dream, right? And 10x has been on the forefront of that for from the industry point, and they just recently announced in one of their I guess webinars or something, it's called, that they will soon, next year, provide an integrated high throughput solution that links antibody to antigen, or B-cell receptor to antigen, and T-cell receptor to antigen. So that will be very nice. But there have also been other papers which I'm citing here. So while generating single cell data is still very, very costly, and unfortunately therefore also quite high throughput, which comes again to the point of how representative of the biology is single cell data. It sounds like a good idea, but how much do we learn from single cell data? I think the jury is a bit out there still, since the data sets are quite low dimensional and mostly focused sometimes on specific subsets of the repertoire. But the software is already there. So we are prepared when the huge data will come in the future. And there are several suits that I'm just mentioning here, from incantation to Skirpy to ImmuneArc, and then also SC Repertoires. So, and they overlap to some degree, but they also have their idiosyncrasies. So just try for yourself, and they're all very good. So as I said, I'm already at the end, because I think one can say a lot about single-cell sequencing, but also, yeah, it will not help you that much. So the main problem with single-cell sequencing is that its throughput is very, very low. So you cannot really profile entire repertoires with single-cell sequencing unless you are super rich. And by super rich, I mean very, very rich. Benchmarking of single-cell sequencing, I think, is still in its infancy. Keep in mind that this Nature Biotech benchmarking paper, this TCR paper, just came out now, and it was on bulk sequencing, showing you how far away we are from actually benchmarking single-cell sequencing data. And still with bulk sequencing data, we are still, as should be also apparent by now, I can still not tell you, you have to use this or this or this for this approach. There's just no real standards yet. And what is also important to know is that most data analysis pipelines, so those pipelines that I'm going to talk about in the next section, are mostly developed for bulk sequencing and cannot be readily used most of the time for single-cell sequencing. So for example, just very simple germline gene usage of VHVL, how would you even represent it? So very, very simple things should be developed at much more, I think. Yeah. Yes, and this is just a summary that bulk sequencing remains the state of the art for very deep coverage. Single-cell sequencing preserves pairing information, and it's therefore superior if you want to really go in depth for a very small portion of the repertoire. Yeah, and there are several approaches, but I think overall we need better insight into how reliable many of these approaches are. Okay, so now we come probably to the section which many of you are looking forward to the most. It's also my favorite section, so that converges well to keep with the vocabulary of this webinar. So we will go through separate sections, which is diversity, network analysis, and machine learning. And as before, I will go quite quickly and only show you brief glimpses of the field. And I think, as with all of my slides, I apologize if I don't show works of some groups or some people. It's just, there's just too much, and I had to make some decisions. But there is much more work to be discovered outside of these references that I show. But the citations that I give you should at least help you to go further and cover the field with respect to the state of the art. So enough of the blah, 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 blah. This is an overview of the computational approaches or questions that you might have and for which computational approaches exist. So you might want to look into diversity, sequence similarity, or repertoire architecture in B, at clonal evolution in C, mostly for B cells, and then clonal overlap, or also called convergence, which can be done both on the entire sequence level or, as shown here, on the k-mer level, on the subsequence level. What I'm not showing here is also a field that has been pushed by the Charlotte Dean lab, is the structural diversity of entire B-cell receptor repertoires, which is on the very lower left of the slide. And there are many papers on this from the Charlotte Dean lab, which I warmly invite you to discover, because it's there are, and there's also another group of which I'm lapsing on the name now, but there are several approaches just to keep it brief, that can infer the structure of the antibody from the sequence at quite high accuracy at very high speed, so that you can look at the structural diversity of entire repertoires, which is very nice. There are many tools for all of these approaches, and I put here the reviews if you want to know more about these tools, and we are going through some of the principles of these approaches and what to look out for. So one of the main things, I think, how the field has started is quantifying and comparing the diversity of immune repertoires, right? We have repertoire 1 and repertoire 2, and we want to say, do they have the same diversity or not? And the problem is, as we have already discussed, low clonal overlap. So it's almost like comparing apples to oranges. And how do we bring apples and oranges together into one new fruit that we can then compare across all the repertoires? And this is what I'm showing here now. So we have the repertoires, right? We have the antibody sequences that we can find here, for example, these 4 CDR3s in each repertoire, and you see that they are different. So you already would panic, right? 0 sequence overlap, what do I do? And then they have different frequencies. So all of this also is, of course, for T cell receptors is the same. So if I don't say things explicitly, most of the things can be used for both BCRs and TCRs. So how can we now compare the repertoires? Is by a diversity indices, which solve the problem by mapping frequency distributions to a common coordinate system. And so what people usually use is the Rényi entropy, or if you take the exponential of this, is the Hill diversity, and different alphas of that, so different parameters map to different diversity indices. All of you might know some of them, maybe species richness, Shannon entropy, Simpson's index, and Berger-Parker index, for example. There are others. What this helps us so keep this alpha in mind, so we can vary this alpha. And if we vary this alpha and compute then the Rényi entropy or Hill diversity, which is the exponential of the Rényi entropy, then we get diversity profile. And what this allows us to do is that it can be shown, which I'm showing briefly in the next slide, that these Hill diversity profiles kind of capture the many characteristics of the underlying clonal frequency distributions that we were not able to compare because the CDR3s were different. The problem with the diversity indices that you hear that many people do not appreciate is that if you only look at one, they can be qualitatively different conclusions depending on where you look. So for example, for alpha equals 1, which is the Shannon entropy, you would say that the blue repertoire is more diverse than the red repertoire, whereas for alpha equals 2, the red repertoire would be more diverse than the blue repertoire. So completely different biological conclusions you would draw if you had just looked only at the Shannon entropy or at the Simpson's index. That's why it's good to check a few indices or look at the profile to see if there's some overlap or some intersection between the samples or not. Then unfortunately, the problem is how do we, and so this Rényi entropy or Hill diversity gives us a number, but what does it actually mean? So there is a very nice interpretation of that. So for example, here we have a repertoire X of 5 sequences, which have a given frequency distribution. And and for alpha equals 1, which is the exponential of the Shannon entropy, we would get 2.28. So what does 2.28 means? It means that if we round down, it's kind of 2. And this would mean that the diversity of repertoire X is equivalent to a repertoire composed of 2 clones with equal frequency. So for example, if we had the repertoire X where one clone would be 99% and the others very little, then the diversity of this repertoire would probably be close to 1, because even if you have 1,000 clones, if one is super highly represented, the diversity is equivalent to just having one clone. So those diversity indices create equivalence classes, which is for the nerds among you, I guess. Yeah, and here we show in this genome medicine paper that clonal frequency distributions here simulated as CIF distributions can be recapitulated by diversity profiles, so which is very nice. The problem with diversity is that since you're undersampling, there is the problem of how true the diversity, or how true the calculated diversity is. Is it a representation of your entire diversity? And for that, you can use estimators. I'm just showing you a few here. I will not go into detail. This would be an entire lecture on its own. All I can say is that I think also here the field is underdeveloped and it would be nice to see some more papers on that to really benchmark different estimators, given also current sequencing depths and clonal diversities. But that should get you started, some of these citations here. So if we look at the clonal convergence, as I said, we can look either on the entire sequence level or on the subsequence level, which I showed there as the black letters, so this DG and this GG. And yes, and there is a very nice review from Castro et al. On public receptors also. So clonal overlap can be by incorporating no frequency. So you just look at the sequence and not how often you find the sequence in your repertoire. This is on the left. And then you can, there's, for example, the Moricita-Horn and versions of that, which includes also the frequency of your sequence. So it weights your overlap by how often a given clone is found in your repertoire. And there are many other approaches that marry these kind of concepts together, and I'm showing you these citations there. I can, if you go to the lower right, the Rempala et al. Journal of Mathematical Biology is a very excellent paper which gives a lot of overview of these different overlap and diversity estimators. One, I think, defining concept in immune receptor repertoires is the concept of generation probability. So what is generation probability? So we all know that if you look at your sequencing data, different V genes are used in different frequencies. So that means it's the probability of seeing a given V gene is different across V genes, and therefore also the probability of seeing certain CDR3s is different across CDR3s. If they are short, they have a higher generation probability. If they are longer, they have a lower generation probability. And all of this can be mathematically modeled by a program that is used, AIRR, which is for the nucleotide sequences, and there is corresponding program which is called OLGA, which is for amino acid sequences. And there is also, so if you just go to these citations that I put on the lower right, you will see also that these frameworks can also include selection, for example. What is nice with AIRR is that once you have trained on your data, you can assign a generation probability to your sequence, which means you can see how likely it was that a certain sequence could have been generated, or is to be generated, which can also then help you understand whether the public clones that you see, is it because they are very easy to generate, or is it because they have been elicited by an antigen response? And though would be a priori unlikely, but this antigen response makes them more likely to appear, which could be used for flagging sequences as antigen associated or even antigen specific, which this whole framework has also been used for. Yes, so this is AIRR. This framework of looking at generation probabilities has also been used for looking at the expected number of public sequences per repertoire in mice and human. And I think also in this paper we have also, I think, TCRs and Bcrs, if I'm not mistaken. And it can also be used as a classifier for predicting whether for a given number of people, a given sequence will be public, so shared among a given number of people or not. So that's very nice. I think an open question here is, do repertoire generation models differ across individuals? And what is the influence of technology on these models? I think there is a bit more work that we could do on these kind of questions. From the diversity point of view, there have also been approaches that have exploited sequence convergence on the subsequence level for predicting antibody antigen TCR epitope binding. So these were two Nature papers from 2017 from Dash et al. And Glanville et al. And the Glanville et al. Paper has now been recently updated with a new with Glyph 2.0 by Huang et al. In Nature Biotech in 2020. Both of these approaches either take into account sequence similarity or K-mer-based similarity to predict epitope binding. What was interesting in these papers was, for me personally, not that very similar sequences could target the same epitope, but that also very different sequences could target the same epitope. And to find those sequences, I think we require completely new methods to find these outliers, which are very interesting to also predict, I guess, in the future. So, as a summary, I guess diversity is one of the hallmark features of adaptive immune repertoires, and it can be quantified using methods from mathematical ecology, because all this Reni entropy stuff has all been done before, counting tigers and dolphins and whatnot. Diversity profiles are superior to single diversity indices when comparing clonal frequency distributions. Diversity can hold also immune informations. I didn't show you that, but it can be used to classify repertoires based on disease state. VDJ recombination statistics can be inferred mathematically and then used to assign generation probabilities to then judge whether public clones are likely to occur or not likely to occur. So the level of surprise that I should have when seeing a public clone. And and repertoire overlap may be quantified from different perspectives: frequency, independent, dependent, and so on. Okay, so the complementary part to diversity is sequence similarity. So because we want to know within a repertoire how similar are those sequences. And for that, people usually use network analysis and to resolve the sequence similarity architecture, as we'd like to say. And I will just point you to one paper, a preprint by Rami Arnaud, where they looked at both sequence similarity and frequency together. Here we will only look at the sequence, not at the frequency of those sequences, most of the time. So how does this work? So you have to build a distance matrix, which can be computationally very expensive. And then you have a distance matrix from which you then build the network, and the network can then be represented with edges and node. The node is the CDR3, and you draw an edge based on the distance that you want. So for example, Levenshtein distance 1 would probably only connect those ones that have similar specificity, for example, if that's your research question. Yes. And what we have seen, and what other people have seen, research groups have seen as well, that is that if you're undergoing an immune response, the sequence similarity architecture of your network looks more like a power law, looks more power law distributed. So you have few nodes to which you have a lot of connections, whereas the Poisson or exponential architecture is mostly seen for naive repertoires. And that can be looked at using the degree distributions, and the degree distributions of a network is, so the degree of a node is the number of connections a given node has. And then you can just count how many times you have CDR3s with a given degree. This may sound very complicated, but it's not. And I put also the papers, I think, yeah, just go to the Miho Nature Communications paper, and this will explain things quite clearly, I think. Yes, and then public clones have been, for T cells, for example, also linked to antigen, so to antigen binding, as was shown by Mahdi et al. In genome research. So there you see that sequences with common antigen binding clustered together. And just as a, if I go back here, do I have that? Oh, yes, here. We also showed that actually public clones are very important for the for your network to keep the architecture of your network intact. If you delete a certain number of public clones from your network, it will completely disintegrate into different clusters and will not have the shape anymore. So, for example, the exponential shape or whatever. So, showing that public clones are usually more connected than non-public clones, which could be due to generation probability. So there you see that generation probability and sequence similarity could go hand in hand. And this is what was also exploited by the Walczak lab, where they used sequence similarity and then looked at whether given neighbors of a given node were more or less likely to occur by chance, using then generation probability approaches. Again, trying to find overrepresented clusters or sequences that could point to antigen binding. Yes, so that was all about network architecture, which can be used to determine antigen recognition breadth if you, for example, link all the sequences that differ by one amino acid. So this is also something I didn't say. Most of the networks, or all of the networks, are based on probably amino acid sequence, since you really want to look at antigen binding. That is not to say that you could also do it, of course, with nucleotide sequences. Yes, and then as of a few years ago, many people only looked at network shape just by eye, but I think we need much more quantitative approaches, such as, for example, looking at the degree distributions and different network measures to compare samples across disease states, for example. Construction of large-scale networks requires still high-performance computing. There are a few approaches now out, which I should have cited here, but I did not. So, for example, there, so for example, there's this classed TCR preprint, which can build very large distance matrices in a very clever way by not being so computationally demanding, for example. Yes, and public clones play a special role in antibody and T-cell networks, which I think we have not fully understood yet. Then coming to phylogenetics, which I have to say I'm not an expert on, so I will go quite quickly. But the main purpose of phylogenetics is to infer the evolutionary relationships from naive B cell to the plasma cell, so the affinity-based evolution over time. And for example, to reconstruct the evolution of broadly neutralizing antibodies could be one application. There are many different approaches that you can use, which are on the left. I will not go into detail. It's explained, and there are also overview articles for that. What one should be careful as well about, because many people just use one approach and then they're happy. We have shown using simulations that phylogenetic approaches may lead to different tree topologies depending on the approaches you choose. So that's something to keep in mind. And as of recently, also trees have mostly disregarded frequency information of the clonal sequence for building the tree. But this has also been addressed, at least to some extent, by the Madsen lab in 2018, by the paper that I'm showing on the lower right. Recent advances in phylogenetic analysis have been to also predict affinity using phylogenetic approaches, again by the Madsen lab on the left-hand side in PLOS Computational Biology. And then the Kleinstein lab, and this is an effort led by Ken Hoehn. So one of the main approaches, or one of the main questions that you have when you look at your tree and you know that you have different cell types in your data set. So for example, naive B cells, germinal center, memory, plasma, you want to see how they stack up in the tree when state switches happen. And here it was an approach was developed that can actually statistically characterize migration, differentiation, and isotype switching along B cell phylogenetic trees to give so that you can say whether some phenomena that you see in this tree are significant or not. So for example, then you can see how often do I see that the memory B cell comes from a germinal center B cell. Is this significant or not? So this is what this package does. It's called Dowser, and it's on Bitbucket, and there's also tutorials for that. And there's a preprint also. So, as a summary again, there exist many methods for phylogenetic inference, but one should be careful because they can lead to different outcome. I think this is not solved right now, and there's also no standardized way of approaching this. But again, I'm not an expert. It might be best to contact these authors that I have just mentioned. Yes. So we will go forward to the last topic, which is machine learning. So machine learning is very, very hot right now, not only in immune receptor analysis, but overall, as you know, from all the high-profile papers. But especially for immune receptors, I think machine learning is really useful to kind of mine the many sequence dependencies that we cannot see by our other computational approaches that machine learning can just learn and see by itself. So there is, just to remind ourselves, supervised machine learning, where you give both the data and also labels of the data, and there is unsupervised machine learning, where you only provide the data, but you don't provide the labels. And then there can be shallow learning. So for example, support vector machines, random forest, and then there are also deep neural networks which can learn abstract feature representations from the data through a hierarchical structure. So many layers. if you want to know more about these things, there I put a review there, and there are many other reviews. I will not say more about machine learning specific. I will more explain what one should be careful about when using machine learning for immune receptor repertoires. So the main, and this is from a review that I invite you all to read, that we just wrote with Lindsay Cowell and Goyari on machine learning in immune receptor data. So if we go to panel C, the main machine learning tasks are sequence-based classification. So you want to, for a given sequence, you want to predict a label, for example, binding to antigen X or Y, and repertoire-based classification, where you want to predict the label of a repertoire. So, for example, autoimmune, like diabetes, yes or no. And there is also a generative model where, based on training, based on some kind of input data, you want to generate new data, new sequences that have that share some kind of property with the input data. So, for example, antigen binding, for discovery of new antibodies. And I think so maybe we go to E, so to panel E. So for machine learning, you always have to encode your data in a certain way. And some of these factors, so you could encode via k-mers, you could encode via Edgeley or Cadera factors, which are physicochemical representations of amino acids. You can use also, as we did previously, you can also use clonal diversity for machine learning. Many things you can use. This is, though, quite similar to normal machine learning, so I will not go into detail. I think this is the more interesting slide, where we kind of detailed the many things that make machine learning on adaptive immune receptor repertoires or sequences especially hard. So one thing is the low clonal overlap, or even a low subsequence overlap across individuals. For one individual, we can have, so now we're at the B, we can have multiple immune states, of course, or for, if we are on the sequence level, also we have polyreactivity, right? So how do we even generate the data to have these kind of information? But even if we had this information, how would we cope with this in a machine learning framework, especially since we know that the disease signal can be quite low. So, for example, for celiac disease, celiac patients have one cell out of 1 million can be associated with celiac disease, for example. So the signal can be very, very low. And even, for example, if in the famous CMV dataset from 2017, only a couple of hundred TCRs were actually associated out of millions with CMV. So the signal is generally very, very low. And you can imagine, if you want to find the signal for many immune states, it can get very tough. And then we go to D. So the diversity of the immune signal is also broad. It can be in the frequency space, so clonal expansion. It can be in the sequence space. It can be also in the 3D structural space with sequence dependencies. There can be confounding factors, which we will briefly discuss, such as age or sex or MHC. And then there are also technological effects that can influence machine learning, of course. And for most of these things, we do not have a real way of dealing with them yet, just as a broad stroke. So to not get your hopes up too much. Here I'm just summarizing a few of the major machine learning works that have been published over the last years. There has been prediction of immune state, prediction of antigen binding, and generative modeling for making new sequences. And we think to benchmark machine learning in the future, and currently also ground truth sequence data generations, such as, for example, using IGO, OLGA, or ImmuneSim, is very important, since most of the time the correct experimental data to benchmark your machine learning approach is not available. This is just very brief, that there's this very nice Sokolova 2009 paper where it summarized how to quantify accuracy for machine learning if you have multiple classes. So, for example, multiple immune states or multiple antigens. And so there's macro accuracies and micro accuracies. I will not go into more detail, but it's good to look into them, especially if your data is unbalanced. As I said, briefly touched upon, these are just a few papers where people have shown that MHC, age, or sex can influence your data. And that, of course, should be taken theoretically into account when you do machine learning. But it's very tough because you either need to stratify your data or using some other wizardry. So it gets very tough. Again, we are at this problem that now we don't have the over-sequencing problem. We have the not enough samples problem here. As I said, in order to be, so whenever I talk to people and many people just tell me, yeah, we just need good data and then we can solve everything using machine learning, that's not really true, because even if I gave people huge amounts of data with all immune states and antigen binding, very, very few would have ready a machine learning framework for a specific research question. That's why I think it's really important to have good simulation suites where you can develop machine learning frameworks now. So when in the future, in, I don't know, 5 years, the really good data comes, we are actually prepared and we don't have to start only then with developing new machine learning approaches or machine learning approaches that are adapted to the research question. Yes, and to also just a quick shout out, we just put out a machine learning platform which helps to standardize your machine learning approaches, which is called ImmuneML. And I will not go into detail. You can see the preprint. It basically allows you to input any kind of data as long as it's kind of AIR compliant. Simulate data, use different representations for your data, use different methods, and to do this in a very reproducible way that is also then shareable across groups. And there's also a graphical user interface, but also a command line for the more experts, and it can be deployed on Google Cloud, etc., etc. So please read that if you haven't already. What I think is really important in machine learning is to perform suitable tests. So you should always test via label shuffling whether your class definition is actually appropriate. So you just shuffle the labels, and then the prediction accuracy should decrease and converge towards the theoretical limit, which is 50-50 for binary, and then for multi-class it can be different, depending also on your balancing of your data. Randomizing sequences is also always nice to just check for any effects that you might have. Equilibration of data sets is quite a good idea. Sometimes, or many times, it's not possible. Testing the effect of undersampling on the classifier is interesting. So we did this, for example, in the ImmuneML preprint. Evaluate feature recovery, which you can do with simulated data, because there you know the signal, so you can see how well a given machine learning approach actually infers those sequences or motifs that should be disease or antigen specific. And then, if possible, stratify your data by age group, HLA, sex, etc., etc., etc. If you know that this is a huge factor for your research question. So to summarize, machine learning is useful for classifying, predicting, and generating immune repertoires. I guess I haven't talked too much about the generation part, but this has mostly been used for antibodies and, for example, Ami Meur and colleagues have shown, so I'm citing the preprint on the previous slide, have shown that you can generate new antibody sequences, for example, based on certain developability criteria. And also Friedensohn and al. Have shown that they can generate new antigen binding sequences based on an appropriate training data set. So machine learning can act on, depending on the encoding on entire sequences or subsequences. We can measure the accuracy of machine learning, but it remains a challenge, and especially I think standardization, reproducibility, and so forth remain challenges so far. I think what it's also a challenge to really extract the important features, because we don't only want a high accuracy, we also want to understand what drives the immune response. And for that, we need to know the underlying features that drive accuracy, and that remains a challenge as well. But that's a machine learning problem. And adjusting for confounders and so forth is also still an issue. And I think it almost also is a question of what is a good training, test and training data. I think this is still very much an open question, especially for antigen binding. Is a good negative data set where sequences are very close but not binding, or very far and not binding? All of these kind of questions, I think, are still out there. Future directions. So I just put some papers here which have a future direction boxes, so you can read them through on your own time. But my personal view is that I think we need better standardization of experimental protocols, so we can actually compare studies across or data sets across studies. We need large-scale antigen annotated data and also disease annotated data. I think, especially for B-cell receptors, sequence and structural modeling should go together. We need a better proteomic understanding of the antibody repertoire. I only touched briefly on this. We need better computational methods for analyzing paired chain data. We need to better be able to interpret machine learning and to better understand biology through machine learning. And we need to better understand also the structure of the antigen-specific motifs that are implicated in antigen binding and immune status. And with that, I would like to thank our many collaborators and my group members that have shaped over the years these slides. And thank you very much for your attention. And I think we are open for questions again.
Excellent. Well, you have lots and lots of questions. Again, to the attendees, we will start at the bottom and work our way up on all of these questions. But if we do not get to yours, do not despair. You will get an answer to your question. It will just be a little bit after the fact. Yes. So let's start with the first one here. Could you comment on long read sequencing technologies for BCR? Are they advanced enough to cover the repertoire accurately?
This is kind of a blind side on my part. I think people have used long read sequencing. I think you still have an issue there with covering the entire diversity, but I would have to. So this is something that I will research more and then put in the written answer, I would say. Yeah.
Okay. Next question. Does a high Libra sequencing score mean higher affinity to that antibody?
I'm not so sure. I think a high score means you have more reads for that score, but I'm also not sure. I would also have to look that up. I'm not I'm not that deep into the Libra-seq technology. Sorry. Yeah. So many detailed questions.
No problem. You get a little chance to do a little research before you answer some of these questions in writing. Next question, I'll put you on the spot yet again. What is the antibody frequency and how is it computed in a repertoire?
So maybe I didn't say this clearly enough. That's true. So what I mean with frequency is always the number of reads that map to a certain sequence. So that's what I mean with frequency. And then, of course, you give an absolute number, and then you can compute the frequency of that. So it's just the number of reads that map to a certain sequence. That's what I mean with frequency. That was not clear. Sorry. Yeah.
We have actually two questions, so I'll combine them, and they're both on visualizing the repertoire. The first was a more general question. What are some good ways to visualize the repertoire? Second question was a little more specific. What is your favorite way, specifically of visualizing TCR repertoire networks?
So the first question, what are some good ways to visualize repertoires? I think it really depends on the research question. So that's why I showed this review where you can either look at, clonal diversity or clonal expansion or sequence similarity. And for all of these things, different approaches are the best. So for example, for sequence similarity, network is the best. For clonal diversity, these diversity indices are the best. So it really depends on the research question. But the images that I showed here are kind of good examples of how one should show the data. So that could maybe be a guideline. For the network question, what are the favorite way of showing networks? That's a good question. So as I said, so if you have small numbers of clones, you can show the network nicely as a picture. But as you have like 10,000 clones, for example, then it's better to just look at the clonal, at the degree distribution, for example. That said, there's a Python package, NetworkX, and there's also an R package, iGraph, which can make networks, and these make nice nice graphs. I think there is another on which I'm blanking also now, which I will also come back to you. There's a better way of showing networks in a nicer way, but I will put this also into the written question. Yeah, I'm blanking on the name now. Yeah.
Next question, is there a software pipeline for the processing of Amplicon sequencing data from raw data to table of counts?
Yeah. So for example, my XCR is used by many, many people. So yes, so you just put in the raw data and you get the table of counts. Yeah. And it's just a few lines. It's very easy. Yeah.
Next question. Would you recommend the best go-to resource for single-cell TCR analysis, especially in combination with transcriptome analysis for beginners?
I think there are not so many. There are only a few papers. And I don't even know the specific papers now, so I would have to look that up. But this field is in its infancy. So there are not many approaches to that, and certainly not many packages that can do a lot in this regard. I think Skirpy does that to some degree, but again, I would have to look that up. Yeah. But it for sure there is, I think, no tutorials for beginners, I think. Yeah.
Okay, good to know. I'll avoid that then. Is there a known mechanism explaining why some error degree distributions generate power laws, maybe somatic hypermutation, or perhaps even sequencing errors?
Why some error degree distribution? Yeah. So I think yes. So I think somatic hypermutation for B-cell receptors is one way of looking at it. For the overall distribution, where you can have also some kind of power law. It depends on how you define power law. It's also based on the generation probability, that some sequences are just more easier to generate and some are much harder. And this is also on a power law. So all of these things are connected to some degree. Yeah.
Next question. In human studies, we do not usually have a high N to really use sex as a variable. Would you comment on how to adjust for sex differences? Is there a set of sex-related genes used in TCR analysis, for example?
Yes, then you're out of luck. We do not know a lot about this. And remember that the sex bias is also MHC related, so it's like a three-way thing. Yes, but I think this is touching on a good point, that in actually to make really good biological conclusions for humans, we need high N. So I see many studies that have low N. They are nice, but the biological conclusiveness is a bit, yeah.
Next question. Is there scope for moving forward with this kind of computational learning to the structural 3D level?
Yeah. So so the machine learning field in general has many approaches for 3D structural learning. I think for now, if we look, that's probably what the person means here, is that if we want to look at the repertoire and use structure also, I think nobody has done that yet. I think to using structural information from entire repertoires for classifying repertoires, I think nobody's done it. But on the sequence level, this is quite common, commonly used by people. Just also for protein-protein interaction, there are many approaches to that. Yeah.
Next question. Regarding antibody-antigen prediction in that area, can some kind of metadata be used with a motif vocabulary to improve antibody-antigen prediction?
Regarding to the antibody, can some kind of metadata be used? Metadata, I'm not sure what is meant here, but I think for antibody-antigen binding prediction in general, we just need much larger data sets. I think metadata, I'm not sure what it could be, but for example, for 3D structural data, we only have, I think, 1,000 non-redundant antibody-antigen crystal structures. We need many, many more to really understand antibody-antigen binding. So better it's to have much more data. Yeah.
Thank you. Next question. Is the almost exclusive focus on CDR3s due to short read lengths problematic for network analysis and ML approaches?
So that's a good question. And so the focus, why we mostly focus on the CDR3 is because there's not so much information in the germline genes, unless you look at somatic hypermutation. But most of the time, most of the diversity is concentrated in the CDR3, and it's the most important for any kind of prediction. Most of the time, if you look at the germline gene side, so the non-CDR3 side, it doesn't give you so much variation across different disease states or antibody-antigen binding. And we have recently also shown that, again, if you look at structural data, the CDR3 is the only obligate, so the CDRH3 is the only obligate region for antibody-antigen binding. All other regions could have, for some structures, have been dropped out, have been not been useful. Yeah. Yeah.
Okay, well, we are running short of time, but we'll try to get a couple more in here. What about using deep learning techniques?
Yes, there are a lot of deep learning has been used. So just go to the overview slide that I have with papers that have used machine learning, and there you will find some deep learning approaches. Yeah.
Okay, last question. Can we use ImmuneML platform to implement some immune-based solution to a non-immune data set?
Yeah, I would not, that would be great, but I think ImmuneML is very immune receptor specific. So I think there are better pipelines for non-immune receptor data. Yeah, as a short answer.
All right. Well, to remind the audience, you will be getting questions to all of these answers. We need to wrap things up. So in concluding, I would like to thank Professor Greif for relating his insights and experience with data processing and analysis of adaptive immune receptor repertoires. I also thank you very much for joining the webcast today. An on-demand version will be available in a couple of days. I'll send a link by email to everybody who registered, and I'll also send a link to the Q&A document when it's ready. Feel free to watch this or any of our on-demand webinars when it's convenient. Thanks again, and have a great rest of your day.
Bye-bye.